All right, this lecture is basically an introduction to innate immunity. So just some of the general concepts and some white blood cells and just some general things to touch upon. So the immune system has multiple functions, very important functions. The first of which is to recognize self versus non-self and threatening versus non-threatening particles. Because if your immune system can't recognize what's threatening against what's not threatening, then it really doesn't have a job, does it? The second function is it's a defense system and it protects against attack by organisms that threaten homeostasis. Homeostasis is just the balance that our body has in order to keep us alive, and it occurs in many different functions, and the immune system being one of them. It's going to produce cells that seek out and destroy foreign antigens while sparing host cells. Now, this is also something very important that goes back to the threatening versus non-threatening concept. All right, if the immune system is out to destroy every single cell in our body, then we won't be alive, and it really doesn't serve much of a function to us. It's more of a harmful thing than a benefit to us. So it's very important that it spares our host cells, and there are actually a lot of autoimmune disorders and a lot of conditions where the immune system fails to recognize that our healthy host cells are actually they're healthy, and it recognizes them as being something harmful. And but that's something we're going to get into much later. For now, just know that it's going to destroy foreign antigens and spare host cells if it's a healthy and properly functioning immune system. And of course, it's responding once again to anything foreign, and foreign things aren't just viruses and bacteria. Yeah, they're organisms, foreign bodies, allergens, dust, pollen, things that are not really bad for us, but the immune system recognizes as bad because it's foreign, and also host tissue, cancerous tissue, cells that have become malignant. So anything of that sort. Now, it's going to approach threats in three ways, and the first way that it's going to approach the threat, or one of the three ways, it's by killing the foreign cells or killing the viruses or bacteria. Infected host cells are destroyed by disrupting their cell membranes or inducing apoptosis. Now, apoptosis is a very important concept when it comes to cell division, cell reproduction, and also in cell immunity. Apoptosis is what we also call in medicine cell-assisted suicide. That's what it is. It's where you force the cell to basically kill itself. So that's going to be one of the ways how it's going to get rid of the foreign object. Another way is by consuming them, or phagocytosis with immune cell activation. And phagocytosis is heavily carried out by your CD4 cells, which are also known as your helper T cells. And this is another star concept when it comes to talking about the immune system, that it's basically going to be eaten up by your immune cells or your white blood cells, which we're going to get into in a little bit. And then, of course, it's going to outnumber them. It can generate enough cells to deal with the hostile stimuli, so you can just think of it as two armies, right? And you have one army with about 20 soldiers in it and another with 200. So obviously, unless those soldiers are multipotent and are very, very strong, they're not going to survive, and it's the same concept here. Some key terms when thinking about innate immunity, just as soon as you hear the words innate immunity, some of the first things that should come into your mind are these terms. It's definitely non-specific, all right? It's not going to specify against any antigen or any foreign body. If it's something that it recognizes that's foreign, it's going to go ahead and attack it regardless of what it is. It's the first line of defense, definitely. It's the first thing that comes into play when we try to protect ourselves, or when our body tries to protect ourselves from anything dangerous or anything foreign. It's immediately available, which is a major advantage. It's there for us all the time, and it's the first thing that's there for us, and it has no memory. So if there's something that the immune system has attacked in the past, it's going to go ahead and attack it the same way if it's exposed to the same thing again, because it has no memory. And memory is something that we're going to be talking about a little later on in the lecture, probably in another upcoming video. But just know for now that it has no memory. And of course, it's antigen independent, which comes into play with no memory and non-specific. That it doesn't matter what kind of antigen you're talking about here, it's going to react the same way to every single foreign body. Now we get into specifics when we go into adaptive immunity, which is coming up later. All right. So again, it's immediately available, so it's present at birth. It kills invading microbes and it activates the adaptive immune system. And it's going to use pattern recognition receptors, which are also known as PRRs, and these are very, very important when we talk about immunology or the immune system. So we have, first of all, pattern recognition receptors are basically, they're going to recognize what's foreign by detecting carbs and lipids on the surface of microorganisms that are not similar to or are different from those on human cells. Now, if you think about it, there are thousands and thousands of carbs and thousands of lipids. All right, so if you're just recognizing the fact that it's a carb and the fact that it's a lipid, you're not going to be a very specific receptor. This is why they're going to recognize in a very broad and general way the presence of microbes. All right, they're just going to let the body know that there are foreign things going around or circulating in the bloodstream. So you have the first kind, which are toll-like receptors. These are the most common, and they can be found on macrophages, on dendritic cells, mast cells especially. And they're going to recognize the fact that there are foreign bodies circulating in the bloodstream. They're going to activate the cytokines, and then the PRRs are going to be activated by the organism against the foreign body. And then you have the pathogen-associated molecular patterns, or the PAMPs, right? And these are present on bacteria, viruses, etc. Basically, they're different than human cells, all right? So the name pretty much gives it away, pathogen-associated molecular patterns. So they're different than human cells. They're associated with the pathogens, and they fit into the MHC, and immune system is activated once they've fit into the MHC molecule, which MHC is also a major concept that we're going to be getting into much later. Okay, so you have some external barriers. You have your intact skin, first of all. Intact skin is very, very important when it comes to protecting your body or protecting your, protecting yourself. Intact skin. If the second there's an open wound, the second there's a cut in your body, you are exposing yourself to thousands of chemicals, to thousands of microbes that can enter and cause infection. So the keratin layer of intact skin is very important when it comes to protecting your body. Then you have your tears, where there's lysozyme present inside the tears, which is going to make sure that any kind of foreign objects are filtered out and they're not allowed into the bloodstream. All right. In your respiratory tract, you're going to have cilia meaning hair-like projections. These are also present on other mucous membranes in the body, and these are very, very important. Basically, they're going to trap anything that's foreign, and they're going to try to get it out. All right. That's why you cough, or that's why you sneeze. The cilia comes into play here. In your stomach or vagina, of course, you have if you're a female in your vagina, you have very, very low pH, and the stomach, for both genders, this applies, very, very low pH. And there are not a lot of things or not a lot of enzymes that can actually survive in such an acidic environment. So if you have microbes, then the low pH is going to be a natural barrier to protect your body from these microbes. And then in your throat and your colon or vagina, of course, you have normal flora. Now, this one you want to be very careful with, because if you have normal flora here, once you become immunocompromised, if you have an HIV patient or if you have a patient on chemo, once they become immunocompromised, their healthy bacteria or their normal flora can actually overtake their system, and they can actually become harmful and less beneficial. And that's known as an opportunistic infection, and that's something you want to be careful about. Opportunistic infections is when your healthy bacteria, your natural bacteria, your normal flora that's supposed to be there takes over because a patient is immunocompromised. So that's just a little keyword to throw in there. All right, so you have leukocytes and macrophages, and these travel through fluid, tissues, and organs via, of course, the two major transportation systems in the body, the lymphatic fluid and the bloodstream. So now it's time for a pop quiz. So in order to rid the body of a foreign body, the immune system uses phagocytosis, which is activated by which of the following? So if you want to go ahead and pause the video right here, you can pause it, answer it on your own, and then you can play it so that I can reveal the answer to you. So if you're going to pause the video, go right ahead. All right, so the answer is CD4, or helper T cells. Helper T cells are the ones that are directly associated with activating phagocytosis. So we have innate immunity. Now, for the purpose of this lecture, other lectures or other professors may want to teach it otherwise, but for the purpose of this lecture, for the most part, leukocytes are synonymous with neutrophils. All right, so leukocytosis is when you have an increase in leukocytes, and leukopenia is when you have a decrease in leukocytes. Leukocytosis is seen with bacterial infections majorly, all right? It's a major, major component of bacterial infections. It's exclusive to bacterial infections. So a spike in your leukocytes, a spike in your neutrophils, think bacterial infection. And of course, it's also associated with the other things that are listed out there, inflammation, stress, malignancy, but it's mostly with bacterial infection. And then leukopenia is where you have a decrease in leukocytes, which is seen in bone marrow failure, chemotherapy, sepsis. And for sepsis, I want to make a note here. For sepsis, it's actually a very poor prognosis if you see leukopenia in sepsis, because sepsis is outspread, just your infection is spread everywhere. If you're seeing leukopenia there, you should actually be expecting an increase in leukocytes if there's a normal immune response going on. But if you see leukopenia there, then it pretty much means that your body's starting to give up, which is why it's a very poor prognosis if you see leukopenia and sepsis. And of course, an autoimmune disease, infiltrative marrow disease. So you have 5 types of white blood cells. You have your granulars and you have your agranulars. And if you look at the pictures, then it becomes very, very easy to recognize which are your granulars and which are your agranulars. But just to list them out here, you have your neutrophils, basophils, eosinophils as your granulars, and then your lymphocytes and monocytes as your agranular white blood. Sorry. All right. You're going to start out with your white blood count all the way to the left, and you're going to see your normal range for white blood counts, for white blood cells should be 5,000 to 10,000. That's your normal range. Males or females, 5,000 to 10,000. Anything out of this range, well, hopefully that should tell you that something abnormal is going on. Now, moving to the top over here, you have your hemoglobin levels. Now, I don't know, maybe I forgot to mention 13 in there, but 13 should definitely be in there. The hemoglobin range varies anywhere between 12 to 15, so that includes 13, all right? 13 is not abnormal. That's actually a small typo. It should be 12 through 15. So 12, 13, 14, and 15 should be your normal hemoglobin levels. If they're out of the range, especially if they're low, then that definitely indicates some sort of an anemia going on in the patient. And then over here to your right, you have your platelet counts. That's 150,000 to 400,000 for your platelet counts. All right, so this should be your normal number of platelets in the blood. And then on the bottom here, you have your hematocrit levels, and your hematocrit is basically the percentage of red blood cells floating around in your bloodstream. And this should be around 35% to 40%. So just knowing these normal ranges and knowing the kind of structure of the CBC with DIF panel is going to make you very, very familiar with what a normal blood count or a complete blood count should look like. Looking down the white blood cells here, you have your neutrophils, eosinophils, basophils, monocytes, and then your lymphocytes. And your neutrophils and your lymphocytes are really the main, I'd say the stars here. All right, these two are the main things that you're going to find in the bloodstream. And your eosinophils, basophils, monocytes, of course, they're there. They're going to make about 10% of the white blood cells in your bloodstream, basophils being the complete lowest. All right, and I'm going to get into each of these individually just in a bit. It's coming up. Remember, we said that it took up to 7 to 14 days to actually form these, and they only existed for 6 hours. All right, so of course you're not going to have enough of these neutrophils to fight off the infection, so they're going to release their baby neutrophils or their bands into the bloodstream, and that's why you're going to see bandemia very frequently associated with neutrophilia. And then you have neutropenia, of course, which is the complete opposite. Those are low levels of your neutrophils, and this is seen star in viral infections. Viral infections are the biggies here. Of course, in aplastic anemia, and as I mentioned before, in sepsis, there's poor prognosis if you're seeing neutropenia. All right, now we have our dendritic cells, or our Langerhans cells, and these are specialized cells in the skin and the mucous membranes, which actively engulf self and non-self molecules. So basically, foreign dying cells and cell fragments, the garbage really, the debris. I always like to remember dendritic cells. It's D for dendritic and D for debris. Debris meaning garbage. So they're going to pick up the garbage, basically. And then we have our eosinophils. Now, this is a major concept, especially if you're going to become somewhere down the road an allergist. This is going to be a major concept in there. Your eosinophils are granulated particles, and these are going to destroy and weaken parasites. And the star here, I don't know why I didn't bold it. I really should have. But the star is that they're going to form allergic reactions. All right. So they're going to phagocytize the antigen-antibody complexes, and they're going to form allergens. They aggregate around large parasites, and they're found in the mucosa of the respiratory, digestive, and urinary tracts. So eosinophils are very, very highly associated with allergies. If there's one thing that you want to remember about them, you want to remember that they're going to form allergic reactions. There is your pretty little eosinophil. You can already tell that these are bilobed, right? You see the two lobes here, the two big purple little thingies, and they stain with acidic dye, and they stain red. Now, that's why they're called eosinophils, apparently. High levels, again, all right, they're going to be associated big with allergies, asthma, eczema. All right, these are big things, and you'll learn later on that asthma and eczema, for the most part, go hand in hand, all right? So they're going to be highly associated with that. Collagen vascular diseases such as they're going to be associated with. Basophils, now these are structurally related to mast cells, so they may go ahead, they may go on to become mast cells even. So just know that basophils are related to mast cells, and they're going to secrete two very, very important things in our bloodstream. The first being a vasodilator, and we haven't really touched upon inflammation yet, but really what happens in inflammation is that your vessels dilate. You have vasodilation so that you can let all the white blood cells into that blood vessel wherever it's needed. And a major vasodilator is histamine. And anticoagulants, coagulation, and we haven't gotten into blood coagulation either, but basically coagulation is where you have platelets just preventing blood loss. All right, that's what coagulation is, where they form a mesh and network that prevent blood loss. And there's a whole coagulation cascade that goes along with blood coagulation that I'm really not going to get into right here because it's going to make things way too complicated. But just know that in the inflammatory process and in the blood coagulation process, basophils play a major role because they're going to release histamine and heparin. They're going to release the two major components of vasodilation and coagulation, respectively. Histamine and heparin. Heparin being an anticoagulant. All right, remember that, anticoagulant. It's going to increase blood flow to affected areas, and this ties in with the inflammation concept. And it's going to be seen in allergic reactions. It's bilobed with granules that stain with basic dye blue. So the way I remember it, pretty easy. B for everything, right? B for basophil, B for bilobed, and B for blue. And immunoglobulin E is going to bind to these mast cells or basophils. We haven't really gotten into the immunoglobulins. It's going to probably be in another video. But for now, just know that IgE is going to bind to these. Monocytes are phagocytic cells that are capable of fighting bacteria and removing necrotic debris. And these are going to produce interferons. Now, interferons are also very, very important when it comes into the inflammatory process, or when we're talking about any kind of foreign body. Because what interferon is, it's basically, suppose you have a building, and suppose that there is a fire in that building in one apartment, right? And suppose you leave that apartment and you're knocking on every single door in that entire building and telling everybody to get out before the fire gets to them. That's what interferon does. Whenever you have a cell that's infected or invaded with a virus or bacteria, it's basically going to go on to the other cells that are healthy and not have been invaded yet, and it's going to tell them that, you know, it's protect yourself because there's a virus in the bloodstream. So that's what interferon does. It's a very, very important communication mechanism in the body. So increased levels, of course, are going to be seen in chronic inflammatory disorders, tuberculosis, parasites, and viral infections, especially in Epstein-Barr virus, which is known to cause mononucleosis. And then you have your lymphocytes. Now keep in mind that you have 3 types of lymphocytes. You have your T cells, you have your B cells, and then you have your natural killer cells. So your T and B cells are not a part of innate immunity. Just remember that. Your T cells and B cells are not a part of innate immunity. So if there's ever a question that's asking you something about innate immunity and they're asking you which cells are involved, remember T cells and B cells are never going to be the answer, because it's natural killer cells that are part of innate immunity. T cells and B cells are actually a part of adaptive immunity. So if they're talking about natural killer cells, it's innate immunity, even though they're all lymphocytes. This can get a little bit tricky, so just make sure you have that concept in mind. They are large agranular non-T, non-B cells, and they attack and lyse host cells that have become cancerous or invaded by virus. And they're going to recognize and kill cells with low levels of MHC1 molecules and antibody-coated cells, or stress-induced proteins, which are released in cells with insult or any kind of transformation or change. And they're also going to produce interferon. Now, lymphocytosis, again, remember, lymphocytosis, high levels of lymphocytes, low levels of neutrophils. So lymphocytosis and neutropenia or leukopenia is going to be highly, highly, highly indicative of a viral infection. Now, it can also be seen in chronic bacterial infections, but viral infections is the biggie here. And also in malignancy. And if you have the opposite, which is lymphocytopenia, meaning low levels of lymphocytes, high levels of neutrophils, so neutrophilia, you're going to have most likely a bacterial infection, and if not, it's going to suggest leukemia, late-stage HIV, and sepsis. So make sure that you have this bacterial, viral, lymphocytosis, neutrophilia kind of a thing. All these concepts, if you want, you should really make a chart of everything and just put it in a chart just so that you know, because it could get a little bit tricky. So you might want to organize this a little better in your notes. Now, going back to that concept, I said it was a very important concept. Going back to that lymphocytosis, neutropenia concept, we're going to talk about the left shift and the right shift. So basically, and this is called a shift, what happens is that when you have high levels of neutrophils or high levels of leukocytes, for the sake of this lecture, high levels of neutrophils, and low levels, I should have really written it here, but it's not written down here, so just make sure that you make a note of this. I'm going to try to go very slowly. High levels of neutrophils or leukocytes, low levels of lymphocytes, so neutrophilia and lymphocytopenia is going to indicate a bacterial infection. And that's why it's called a shift to the left or a left shift, because you're going to have high levels of neutrophils, all right? So remember we said that neutrophils were 60% and lymphocytes we said were 30%. Well, now imagine that your neutrophils are at a 90% and your lymphocytes are at 10%. So imagine that your neutrophils are to the left, your lymphocytes are to the right. And your neutrophils are raised. They're at 90%. Your lymphocytes are down. They're at 10%. So you have neutrophilia and lymphocytopenia. That's shifting to the left because the left side is raised. So you have bacterial infection going on in the body. Now the opposite of that is when, say, you have 90%, which is very rare, not very common, but for the sake of the matter, 90% of a raise in lymphocytes. So now you have lymphocytosis. And what's going on the left side? Well, the left side is going down, definitely. So you have neutropenia or leukocytopenia. So on the right side now, you're having an increase in your lymphocytes, a decrease in your neutrophils or your leukocytes. And that is going to be highly indicative of a viral infection. Now you're shifting to the right. So make sure you really do grasp that concept, because it's going to be very, very important in most immunity or immunology classes, I believe. If you have any other questions, I'm going to try to put my email address in the description box below. Feel free to email me if you have any other questions, and there are definitely more lectures to follow.